Well, good afternoon, everybody. I'm Donna Caffell. I am the eScience Projects Coordinator at the University of Massachusetts Medical School. And I am delighted to introduce to you our next presenter, Dr. Jane Fraser. Dr. Fraser is the Robert M. and Shirley S. Siff Chair in Autism and Professor of Psychiatry and Pediatrics at the University of Massachusetts Medical School. She is also Vice Chair and Director of Child and Adolescent Psychiatry, has a faculty appointment at the UMass Graduate School of Biomedical Sciences, and is the co-director of the Child and Adolescent Neuro Development Initiative, which is often referred to by the acronym CANDY. The CANDY Research Program is part of the Child and Adolescent Division of the Department of Psychiatry at University of Massachusetts Medical School. As co-director of CANDY, Dr. Fraser directs a broad research program that addresses a variety of scientific problems relevant to advancing the understanding, diagnosis, and treatment of individuals with neurodevelopmental disorders, such as early onset bipolar disorder, schizophrenia, and autism. The CANDY team studies all three diagnostic groups since there is often overlap in the clinical symptoms, genetics, and the neuroimaging findings of these disorders and in the clinical interventions used to treat them. CANDY is involved in a variety of research pursuits, including genetic studies, intervention studies, and neuroimaging efforts. Dr. Fraser collaborates with a number of investigators, both locally and nationally, in order to tackle difficult questions. The neuroimaging studies use technologies such as magnetic resonance imaging, commonly known as MRIs, to investigate the development of brain structure and function. Using these techniques, Dr. Fraser's team hopes to characterize normal development and advance the understanding of issues surrounding mental illness and atypical development in their interventions. The Child and Adolescent Neurodevelopment Initiative at UMass Medical School is currently involved in clinical research in early onset bipolar disorder, psychotic disorders, and autism spectrum disorders. The research team is led by Dr. Fraser and is supported by a group of professionals, including child psychiatrists, advanced practice nurses, neuropsychologists, statisticians, research coordinators, and other interested volunteers. This research program is part of the Child and Adolescent Division of the Department of Psychiatry and attracts many individuals from the greater Massachusetts area as well as throughout New England. Several of CANDY's collaborations are national and international in scope. So I would like very much to give a warm welcome to Dr. Jane Fraser.
It's on now. Okay. It's a delight to be here. I have to share something with you. I was telling Elaine and Donna earlier that throughout my schooling, one of my favorite places to be was in the library. And I actually, throughout my schooling, either volunteered or worked in libraries. And I actually thought I would become a librarian one day. And it's such an honor for me to be here with this esteemed crowd. And when Elaine asked me, I was just delighted to participate. I also am honored to follow Dr. Monaco because I think he did a beautiful job of outlining the issues as it pertains to genetics and neurodevelopmental disorders. And happily, his talk and my talk nicely complement each other. So I am going to be talking about neuroimaging of children's brains. This is a work that I've been engaged in since 1993. I did my first research actually in the intramural child psychiatry branch at National Institute of Mental Health. I worked for a number of years with Dr. Judith Rappaport there, and then returned to the Boston area initially, where I worked in the Harvard systems and continued to do my neuroimaging, and have been now at UMass for 4 years, and it's a pleasure to be a part of the UMass community. So I'd like to start by outlining the objectives of my talk, which is I really want to explore with you the use of neuroimaging techniques and how we apply them to the living brain. because neuroimaging actually can provide some valuable insights in terms of potential biomarkers for illnesses such as childhood onset neuropsychiatric or neurodevelopmental disorders. So to start, I'm going to talk a little bit about typical brain development and also talk to you specifically about the various neuroimaging techniques that we employ in neurology and psychiatry, and highlight the techniques that we use specifically to work with children who are suffering from various ailments. And then we'll talk about brain development gone awry. I'd like to also then talk to you about the use of biomarkers and what biomarkers are and how we can employ them in the work that we're doing. And then I'm going to end by talking about autism as an example of the kind of research that actually can be done in our clinics. So let's start with this. These are two scientists, and they're looking from point A to point B and looking in the middle. And one scientist says, I think you should be more explicit here in step 2. And that's where then a miracle occurs. This really does describe what it's like doing neuroscience. It's very complex. As you might imagine, the brain is a very complicated organ. And we are gaining some insights, but we still have a lot of work to do. Now, I thought I'd put this in here because this has a variety of components to it. If you look here, this is actually a drawing done by Leonardo da Vinci. Many years ago, people used to believe that once we were born, the brain was its full size, and that's not the case. This is actually a little caricature that looks at what happens to brain growth after the neural tube closes, and you can just follow your eye around in kind of a circular fashion until we get to the adult brain there. Again, this is another schematic going from 25 weeks, and this is a human brain, 30 weeks, 33 weeks term, and then the adult brain. So there are some basic principles about brain development that I do want to highlight for you just as a background. It's a very exciting area of research, and actually I'm partial to the work that I do, and I feel like it's probably the most valuable area of research because much of what we learn from brain development in utero and early on after birth translates into what happens in adulthood. So first of all, some basic principles that I think might be valuable. Most brain structures start small and then they get big, all right? And then they get small again. And that's a really basic principle. And I'm going to use the frontal gray matter here. This is actually a cartoon of some data that was acquired partially while I was in the intramural working with Dr. Rappaport and Jay Geed. They continued acquiring data, but this is normal brain growth data on healthy children. And this line represents boys, this line represents girls, and it goes from age 4 to about 21. And it's this period of time, this age range, that's actually pretty important. And you'll see this table again later in the talk, but I use this as an illustration of this principle, which is brains, this is the frontal gray. Frontal gray matter starts small, gets big during this time frame, and then gets small again. And you see the same pattern in the boys. So we call this period of time of brain growth really kind of exuberance. And then at this period of time in development, there's pruning that goes on. It's similar to the terms that we use actually in gardening. You know, there's exuberance and arborization that occurs in the brain. And if you think of a tree or a bush, you know, they grow, they exuberate, if you will, or they arborate, and then we take our little pruning tools and prune back. And it's the pruning that's really critical in brain development processes. You know, there is development that goes on in the brain in utero, and it continues onward into adulthood, and that's very important to realize. So there's exuberant connections and this pruning is critical. And again, this time frame from about age 8 until 14 is a critical time frame. It's a time frame when all these processes are going on and it's complicated. And as you might imagine, the more complicated a period of time there is, the more potential that something can go awry. Okay? If there are too many things going on at one time, there's more potential that something can go awry during that time frame. And again, this is a period of time when kids are experiencing pubertal development. It's a period of time, and again, I'll highlight this later, when most psychiatric disorders onset in children. Now, the exact timing of this does vary somewhat, but pruning, this process of pruning, is a process that actually makes the brain more efficient. Basically, the brain whittles back synapses and dendrites that it doesn't use. So it's guided by the use or lose principle. And the exact timing varies by structure, and not every structure in the brain goes through this exuberance and pruning back at the same time. Now this is the neuroscience interaction triangle. Again, this dovetails nicely with Dr. Monaco's talk. He talked a lot about genetics, but Dr. Tom Insel, who is in charge of the National Institute of Mental Health, is really interested in the concept of how does what goes on genetically translate into what's going on in the brain, okay? And then how does that translate into what's going on in terms of behavior? Now, some of you earlier asked this question about also the interaction with environment, and that's critically important in terms of the field of epigenetics and de novo genetic findings. So in imaging, particularly, we have the capacity to look at all of these issues, and sometimes genetics is paired with neuroimaging studies, but typically in neuroimaging studies we look at the anatomic structure, function, connections, and systems, and we also look at the associations of any abnormalities with behavior, such as mood dysregulation, reaction time, and other such things. Now, my colleague, Dr. David Kennedy, who is really the brains behind our operation, loves this slide, and I thought I would put this in there in honor of him. And it is another timeline for you to take a look at, but I think the important take-home points here is, you know, we're interested in really what goes on in the brain from conception forward. There are a number of processes that occur in utero here. And then the child is born, and subsequent processes happen like myelination, synaptogenesis, and apoptosis. But the most important point of this slide is that much of what goes on, even in utero, translates into behavior or abnormal behavior on into adulthood. And this is what we're most interested in looking at, the association between what happens early on in the brain and how it translates into what we see in adolescence and adulthood. Now, just in terms of basic timelines and brain development, we're going to focus mostly on what happens from birth onwards. Now, from birth to age 3, there's a rapid time of intellectual and emotional and physical growth in children. And in terms of brain and brain wiring, there's a robust, rapid growth. And then by age 6, actually, the brain achieves about 95% of the brain development or adult brain size. It's completed at that point. But there are other processes that continue to happen beyond age 6. Now, I talked a fair amount already about this time frame, the preteen time frame, from 10, or I would extend it downward to 8, up until about 12, 13. There's a second major brain growth spurt that occurs during that time frame. Okay, so the brain becomes yet again very vulnerable during this time frame. It's particularly vulnerable during the birth to 3 time frame, and then becomes vulnerable again because of this second major brain growth spurt. Now in adolescence, from age 13 to 20, this is when a lot of the pruning and refining and organizing goes on, and that's when the frontal lobe actually comes online. Okay, the frontal lobe is really responsible for keeping our emotions in check, helping us to pay attention, and it's not fully online until later adolescence or early adulthood. And that's why we've got a number of neuroimaging studies actually that substantiate with what we all tend to know. How many of you are parents in this room, by the way? Okay. How many of you have adolescents in your household? Okay. So when you look at your adolescent, what do you expect from them when you look at them? So do you see physically almost an adult being? Okay. Do you sometimes expect them to think the way you do? And how often do they? Very rarely. And in part, it's because they don't have a fully developed frontal lobe. You know, they're pretty affectively reactive to things. They don't pay sufficient attention, and that partly is a reflection of the fact that they don't have the wiring to behave like we do. Okay. Now I'd like to talk a little bit about the employment of brain imaging, because I know that was a real interest or a stated interest amongst the crowd. This is such a cool technique or set of techniques because, you know, obviously we can't do brain cuttings, right? But we have a window of opportunity to understand what's going on in the living human brain. And what we have the capacity to do is to look at the structure of the brain. That's allows us to actually use some of the physical properties of the brain to look at the structure of various areas in the brain. We also have the capacity to look at function, and this is really functional neuroimaging. It takes advantage of some of the properties of neural communication, which is really cool. And then we have the capacity to look at metabolism, and that really rests on using some of the chemical properties of the brain, such as neurotransmitters and amino acids. Now, we have the capacity to actually evaluate the neural activity that goes on in the brain, and that harnesses kind of the biophysical effects of the brain. That's why I need people like David Kennedy, who's a physicist, to help me out. We can measure things that require energy, for example. There's a technique called PET scanning, which I'll explain in a little bit, but that measures glucose in the brain. We also can look at blood flow in the brain by using PET or SPECT or functional magnetic resonance imaging. And there are other techniques that allow us to actually look at nerve impulses, which is also cool. And so, for example, we can look at electric fields using EEG. We can look at magnetic fields using magnetoencephalography. And I will tell you about these various techniques as we move along. So PET is actually really an interesting technique, and it allows us to actually use radio-labeled items like glucose and inject it into the brain and then trace it as it goes through the brain. Or we also can use fluorine. Single positive emission computed tomography, or SPECT, also uses radioactively labeled items and then gets a picture of the brain that appears something like this. Now, I just wanted to give you a brief introduction to these techniques, but it's important for you to know that we typically don't use these techniques in children because they're invasive, okay? but it is a wonderful technique and is typically used in adults. But on the next slide, I want to show you two techniques that are being employed fairly readily in children. One is the electroencephalography, and the other is magnetic encephalography. This is the MEG scanner, and this is a pictorial representation of what happens in using the EEG. And these two techniques use intrinsic electrical and magnetic signals from neural currents and conduction. And I'm going to show you just briefly a little video of brain activity. And this is acquired in the MEG scanner while a child is reading. Okay. Now, the common technique that we use specifically in child research is magnetic resonance imaging, or MRI. It really employs a large magnetic field and uses radio frequency and evaluates the intrinsic protons in the brain and then produces a picture like this. And as you can imagine, it's actually some of these open bore magnets like this are very comfortable for kids. And we often allow parents to go into the imaging suite with them because they like to have their parents holding their leg, which is quite comforting. But it gives a really pretty picture there of the brain, and we get a sense of the structure of the brain. Now, what can MRI tell us? Well, the nice thing about MRI are there are multiple components of it. We typically in our lab do all of these. We look at the structure, which this is a picture of some of the color-coded structures in the brain. We look at metabolites, and I'll talk about each one of these individually, but this is a graphic representation of the chemical spectra that we can acquire from certain areas of the brain. We can also look at blood flow, and this is a picture of evaluating blood flow in the brain. And then a more recent technique, which is really quite exciting, is looking at the white matter connections, because we historically did not have the capacity to fully evaluate white matter in the way that we currently do. And this is a technique called tractography. So magnetic resonance spectroscopy is a technique that we use quite a bit in our lab, and at the end of the talk, I'll share some data that we've acquired from children with autism. But MRS, it's non-invasive. It's an analytical method to measure chemicals within various body parts. We're focused in our lab on the brain specifically. And it works based on the physical characteristics of certain elements that are actually visible to the MRI machine, such as hydrogen, phosphorus, carbon, oxygen, lithium, and fluorine. Now, I want to talk to you now about how the brain really looks in typical development. Our lab has recently been involved in, I know you're interested in collaboration. We do a lot in the way of collaboration. I have to tell you, the disorders that we study really require collaboration. The sum is larger than the individual parts. We went through a decade and a half ago, the decade of the brain, and a lot of NIH funding was given out to various principal investigators who did very good work. But you can't acquire sufficient power at one site to really fully understand what's going on in normal development, let alone disease. Okay? And so more recently, we've been partnering with people to have a multi-site acquisition on the one hand. So for example, President Obama released 2.5, 3 years ago now, the ERA grants, the stimulus package grants to get people engaged in research. And we partnered with 8 other sites to make an application for an ERA grant to acquire genetic material, brain imaging, and cognitive functioning in normal children from age 3 up until 21. Because if we are going to fully appreciate what's going wrong in brains, we have to understand what's going right in brains first. So we've collected this huge normal database that's going to be made publicly available. The other thing that David and I are funded to do, which is really cool and I think speaks to some of the questions you asked earlier, historically in imaging and genetics work, people used to hold their cards pretty close to their chest, and there wasn't a lot of sharing that went on. But the NIH is all about data sharing now. And David and I just completed our first 3 years of a grant where we're trying to be an example to the field, where we're releasing all of the scans that we've acquired in the past 15 years into the public domain. So we've actually created a portal to do that. And happily, well, knock on wood, we'll see what happens, but we just submitted our renewal, and it was just reviewed, and it actually did quite well in study section, so we may have another 5 years to continue that effort. At any rate, so a lot of our work is looking at normal brain development, again, to provide a foundation for understanding how the brain goes awry. So let me just touch upon a few points here to let you know what brain imaging has already shown us. Just look at these 3 brains. This is a child at 3 months, a child at 6 months, and a child at 12 months. Can somebody describe what you see? More white matter. Yep, exactly. And also you see a little less gray matter, right? And down below here is just kind of a color-coded example of the same imaging slice. Now this is brain development also across time. This is a larger span of time, but going from 2 weeks, 1 year to 2 years. And this is specifically looking at the white matter. Again, I told you historically we didn't have capacity to really understand where a track is going from point to point in the brain. But with the advent of tractography, we have the capacity now to trace those connections in the brain, which is very exciting. So now I want to turn to children and adolescents. This is using the data from the NIH study that I talked about earlier. This is again that graphic of the frontal gray matter demonstrating going from small to large to small, demonstrating the processes of exuberance and pruning in boys and also girls. And here we have a similar graphic looking at white matter. And what you see during this time frame is that there isn't pruning. In fact, the white matter is increasing, which is an indication that the connections and myelination are increasing over this age range. This also is an example of some of the work that's come out of the NIH from Judy Rappaport's lab. This is a novel technique that was established by Dr. Thompson in California. Judy shared their data with him, and he looked at cortical development over the time frame between 5 and 20 in a group of youth. There were hundreds of children in this data set. And what you can see is, if you look at the areas here in red, red represents more gray matter in the cortex. And as you go forward over time, you'll see it becoming more blue, okay? And what that means is there's more gray matter, there's kind of redundancy here, and then over time there's a pruning process that occurs where there's less gray matter and the brain becomes more efficient. Now I'm going to show you a video that shows this back to front wave phenomenon of pruning over time within a matter of 15 seconds. So turning again to other examples of typical development, ping is the name of our ERA grant. And these are the data that we've already acquired. Our goal is to acquire 1,400 images of youth between the ages of 3 and 21. But this is the first pass of data analysis across that time frame, looking at total brain surface area, left cerebral white matter volume, and performance on a vocabulary test over that same time frame. And again, the same principles. You'll see small, big, and small. And then with the white matter, you'll see growth over this time frame and then leveling off. And vocabulary increases over that time frame during these brain growth. And specifically, you'll see robust increase in vocabulary in this time frame from about 10 to 14, again, overlapping with a period of time when there are multiple processes going on in brain development. So that was a snapshot of typical development, but what happens when something goes wrong, right? Let's look at what happens in a brain image when something goes wrong. Now, this is an extreme example of atypical development. These actually are adult scans, but I wanted to put these in for you so that you can see that structural imaging allows us to see really bad things, okay? So there's an individual who has had a stroke, and then there's an individual here that has a glioma. But the story is different when it comes to looking for brain atypicalities in other disorders like childhood onset neurodevelopmental disorders. Again, this is an Alzheimer's disease. There's a control. This is an individual with somewhat milder disease, and this is a progression from mild to very significant Alzheimer's disease. There's a color coded picture here looking at normal brain activity, mild cognitive impairment, and then at somebody with Alzheimer's disease. Again, this is a pretty significant stage of disease, and you can see a lot on the anatomic scanning. Now, let's turn to neurodevelopmental disorders. These are scans that were acquired within our lab. These are 3 16-year-olds. Do those brains look any different from one another to you? Yeah, the truth is the white matter's a little more brighter in this one, but the fact is if we were to measure them, various brain areas, there really isn't any difference between any of these scans. But let me show you what these individuals suffer from. This is a young man with autism, this is a young man with bipolar, and this is a healthy control. So the brain abnormalities don't leap out at you like the glioma did. Okay. So even when the images look similar, there are lots of differences between patients and control groups. And what we need to do is use our imaging technology to look deeper and deeper, okay? So Dr. Monaco talked about looking at genetics and then looking at the cellular level in the brain. Imaging doesn't give us the capacity to look at the cellular level of the brain. Brain cuttings allow for that. But imaging allows us to look at the brain in vivo, and by using techniques like diffusion tensor imaging and functional imaging and MRS, we can look deeper into the brain. So I now want to talk to you about when things do go awry. Again, you recognize this graph here of the frontal gray matter development. I talked to you about this age span, and I talked to you about this being a critical time when many things are happening in terms of brain growth, making it a vulnerable time frame for the onset of disorders like ADHD, like anxiety disorders, like mood disorders and schizophrenia. And this demonstrates the kind of average age of onset of all these disorders, and there's a really nice overlap that occurs during this time frame. So at this point, I'd like to talk to you about what we're learning about the brain using imaging by studying children who have various disorders. First of all, this is looking at anxiety disorders, and this is looking at individuals with major depressive disorders. These are all teenagers, and typically in imaging, one thing that I get asked often is, can you use neuroimaging to make a diagnosis? Okay, even clinicians ask me that, because everybody wants that. Everybody wants the litmus test. You know, let's have the litmus test in psychiatry or in pediatrics. The fact is, we're not there yet. Okay, so when we do studies, we compare groups to each other, and usually we have to have somewhere, depending on the technique, somewhere between 10 to 30 in each group to have sufficient power to inform us about brain differences. Okay. Now this is functional imaging, and we actually, these youth were exposed to an afraid face paradigm versus a passive or control paradigm to look at where children with anxiety disorders and major depression light up the brain a little more than controls. The amygdala is a structure that governs our ability to modulate our affect. And in both of these disorders, the amygdala lights up much more in those who suffer from anxiety and depression when they are compared to the control group. Now this is diffusion connectivity, and this also is a study that we completed in our lab. We've been very involved in trying to understand the pathophysiology of early onset bipolar disorder, which many of you know is a topic of great debate in the literature and great debate in the public arena. The fact is, children do suffer from bipolar. Okay? It's not as common as it occurs in adulthood. There are very few children who have prepubertal bipolar disorder, but they do exist. Usually the onset is more in the teenage years. And we compared youth who had bipolar disorder to healthy controls, and I'll try to explain this a little bit to you. This is an example of the use of DTI, or diffusion tensor imaging, which gives us some knowledge about the white matter connections in the brain. And we contrasted a cohort of kids with bipolar to healthy controls, and what we found basically is that they had abnormal connectivity in their brains compared to the healthy controls in a number of white matter tracts. So here we have the cingulum, paracingulum, where they're abnormal. The frontal orbital cortex, they have abnormal connectivity, and the cingulum, they had abnormal connectivity, as well as the superior longitudinal fasciculus. The cingulum, paracingulum, and FOC are actually important in terms of their connections because they're involved in affect regulation. So this might in part explain why these youth are suffering from mood swings. And the cingulum and SLF1 are involved actually in attentional capacity. And for those of you who know individuals who have bipolar or youth even who have depression, you know that they typically have some difficulty, even when their mood is stable, paying attention. Okay? And so this might explain why they're having those difficulties. Now let's turn to the application of imaging to autism. This is actually a paper that was published by Martha Herbert, who's a long-standing collaborator of mine at Mass General Hospital. She published this work in 2004. It's an elegant piece of work, and I'm going to try and explain it to you. It's based on anatomic imaging. And basically, there's an analytic technique where they outline various structures of the brain, and then they color code them during the analysis. But what Martha found in this particular study is that children with autism have much more white matter. It's larger, okay? And she found that the white matter areas that are more peripheral are much larger, and those that are deeper are affected less so. And they did a number of analyses that actually gave us some clues into critical periods of prenatal and postnatal white matter development. Again, I'll try to explain this to you as best I can because it is complicated. First of all, this is a graphic representation separating out the superficial white matter and looking at the deep white matter. And as you can see, there are more significant differences in the superficial radiata. Now, they also looked at, and this is really a compelling example of how you can use the scans acquired and then look at the data in a very detailed way. Remember that early on I told you about how prenatally there's a period of time when stuff goes on in the brain that really could have an impact on what's going on in adulthood. What they did in this particular study is Dr. Cavanis and Dr. Herbert looked at various white matter areas and determined when myelination starts in those white matter tracts based on mouse and chimp data predominantly, and also some human brain cuttings. And then they looked at those tracts and determined which tracts were most affected, and found that in autism, the tracts that had the start of their myelination at 45 weeks from conception were the most significantly impacted, okay? So if children are born at 36 to 39 weeks, this is just after birth. The other thing that they did is they looked at presence of mature myelin, and I'll go to the next overlay slide here. They looked at the mean maturity interval and wanted to know if the length of maturity interval had any bearing on the tracts that are most affected. So the mean maturity interval consists of time of conception, time of onset of myelination in certain tracts to the endpoint of myelination. And what they found was that if the longer the mean maturity interval, the more significantly affected are those white matter tracts. All right, so what is a biomarker? A biomarker is a distinct characteristic that is an indicator of a particular biologic condition or process. A biomarker potentially can aid us with diagnosis. It can be used to monitor therapy. And the types of differences we just saw are candidate biomarkers. So in other words, if there's an abnormality in terms of blood flow in the amygdala in depression or bipolar disorder, that's a potential biomarker. But the question becomes, are these biomarkers that reflect the state of the disorder? Are they biomarkers that reflect the genetic predisposition and risk factor? Or are they biomarkers of not the disorder itself, but some domain of dysfunction, all right, that crosses diagnostic categories? So, for example, Tom Insel is really all about looking at domains of dysfunction. And that's really critical, because if you think back to something that Dr. Monaco said, it's pertinent to illustrate this point. He talked a lot about certain elements, genetic findings, that are similar between autism, bipolar, and schizophrenia. Remember that? Okay. Well, no wonder we haven't found the genetic underpinning to any particular disorder if there's overlap in those genetic findings, right? So the current thinking amongst many people who do clinical genetic studies is that maybe those genetic findings, rather than being tightly associated with the disorder, maybe they're more tightly associated with a domain of dysfunction, like executive functioning problems, or in the case of autism and schizophrenia, if you think about those two disorders, Dr. Monaco explained autism very nicely in terms of its characteristics, but for any of you who know somebody who suffers from autism, they have difficulty relating to other people. That's a core deficit. It's not that they don't relate to other people, but they relate in various ways that are different from somebody who's neurotypical. And they tend to be somewhat socially withdrawn. If you think about individuals who have schizophrenia, okay, they suffer from things like hallucinations, delusions, but they also have what we call negative symptoms, and they have difficulty with being motivated. They have difficulty enjoying things. They have difficulty speaking, so they suffer from alogia. But one of the negative symptoms is social withdrawal. Okay? So could these genetic similarities seen be more tightly associated with things like social withdrawal? I guess I have a few more minutes, so I want to try and get through some of this. So, autism spectrum disorders, as Dr. Monaco highlighted, are becoming more and more prevalent. In fact, the CDC just released its recent data, and it's affecting 1 out of 88 children. And about every year, it increases by 20%. So it's important that we do this work. It's important that we understand what's going on in the brain. We have a number of pharmacologic agents available, but those agents treat behavior. They do not treat the core symptoms of autism, and we've got to get better at our interventions. That's why we have to use mouse models, and we have to use imaging to inform the treatments that we deploy. So in imaging, we can look at biomarkers. What can we look at? I said earlier that we can look at things like neurotransmitters. We also can look at things like glutamate. Glutamate is an excitatory neurotransmitter, and Gaba is the main inhibitory neurotransmitter in the brain. And when there's an unequal balance or disequilibrium between these neurotransmitters, it often results in some sort of behavioral dysregulation. Magnetic resonance spectroscopy we use to actually measure a number of brain metabolites, but it does measure things like glutamate, glutamine, and GABA, and provides a useful way to measure biochemical information at a local level within many brain regions. Again, this is a spectra that's typically acquired from an area in the brain. We, in our lab, usually look at the anterior cingulate, which is in this box here. And one of the peaks that we're most interested in is GABA. And GABA is actually harder to find in the spectra because it's hidden behind another peak. But we do a special editing technique that allows us to more fully evaluate GABA, which is the inhibitory neurotransmitter in the brain. Again, this is a nice cartoon that shows how a disequilibrium between this excitatory and inhibitory neurotransmitter can result in psychopathology. Now, Dr. Monaco talked briefly about animal models that allow us to look deeper at the molecular level and give us some clues about autism spectrum disorder. He talked about Fragile X. He talked about tuberous sclerosis. My lab is actually very much involved in those clinical trials that he talked about. And the nice thing is I think we're using science to get closer to finding ways to help people in real life. So there are 2 models of Fragile X that result in the same phenotype. This is looking at 2 mouse models for autism, Fragile X, and also the tuberous sclerosis mouse model. In the tuberous sclerosis mouse model, they make too little of a certain protein. In Fragile X, they make too much. And the goal is to correct the problem so that they represent more of the wild type or healthy mouse. All right, so we're going to talk just briefly about the foundation for our pilot project, and we are interested in glutamate and GABA. I have very preliminary pilot data to show how we're using MR spectroscopy more recently. But there are glutamate and GABA abnormalities in autism, and they've been found both in the brain. There's too much glutamate and less GABA. Genetic studies have substantiated abnormalities. We've also found abnormalities in the plasma of individuals with autism spectrum disorder. GABA is also dysfunctional in ASD, and this has been substantiated by numerous investigators using a variety of techniques, including PET, SPECT imaging, MRS, and genetic studies, hearkening back to some of what Dr. Monaco said, is there are microduplications of chromosome 15q11 and 13, which he did mention in his talk. It's one of the most common genetic deficits in ASD, and there are multiple GABA receptor subtype genes in this particular region, so that's an important genetic finding. So we had a number of questions to answer in our pilot project, and again, I have just a few more slides because I want to just give you a taste of our current work. The questions to answer are: the findings in genetic and animal studies have led to the development of some new drugs focusing on glutamatergic and GABAergic systems, and again, we're involved in those clinical trials. The underlying molecular differences could explain the different response to treatments, so that's really important. If a child with ASD who has high glutamate, there may be some who have high glutamate, there may be some who have low GABA, and those two groups may respond differently to different medications. So we might be able to use biomarkers as predictor of treatment response, for example. There are biomarkers for CNS glutamate and GABA dysfunction that seem to correlate with deficits in autism spectrum disorders that can be studied in a non-invasive way. And do these biomarkers predict the response to the available glutamate and GABA modulating therapies? And do these biomarkers change and correlate with therapeutic response? So in our pilot work, we're using MR spectroscopy to look at teenage boys. We're focusing on boys specifically because autism is affects more boys in a ratio of 3 to 4 to 1, as Dr. Monaco also said. The other reason is it's a small pilot, so we want to have a sufficient M to show a difference. And girls actually have fluctuating GABA levels during menstruation, so we wanted to eliminate that confound in this particular study. We are looking and focusing particularly at the anterior cingulate here, and why the ACC? Because it's a key component to the limbic system. It's implicated in monitoring social interactions, theory of mind, joint attention, and repetitive and stereotypic behaviors, which we see in individuals with autism. It's involved in helping us plan, initiate, and self-monitor goal-directed activities. Okay. So we have 3 goals of this study: to quantify differences in GABA and glutamate neurotransmitter levels in this area of the brain and compare individuals with ASD to healthy controls. We also want to correlate the severity of social communication with these levels in the ACC, and we want to correlate functional imaging response to emotional phases with GABA and glutamate levels in the ACC. Now, at the time that I put these slides together, again, this is just a taste of what we're doing, but I wanted to give you a sense of what we're finding already. Obviously, we don't have sufficient power. As of today, we have 4 and 5 in each group, but we put this slide together when we only had 2 youth with ASD and 4 healthy controls. As you can see, the glutamate levels, there's a greater standard deviation normatively in this age range. There's a less of a standard deviation, more consistency, and glutamate levels were statistically significantly higher in these 2 individuals with ASD compared to the healthy controls. Now, we also looked at GABA and there is no statistically significant difference as of yet, but we're continuing to acquire scans on youth, and hopefully we'll have more findings in the near future. So in conclusion, what have I shared with you? We've examined the specifics of neuroimaging techniques and how they help us to understand typical development. Also, they help us understand what's gone awry in brains. They're helping us to identify candidate biomarkers. And we've really identified some of those actual biomarkers in autism spectrum disorders. But it's a long road for us to hoe here. You know, we need a lot of subjects. We need the power of collaboration. And this is all to be continued. And with that, I'd like to thank all of the people who work in my lab. Again, even at the local level, we have an army, and everybody here keeps me honest and in line and off the streets. And they're all really committed to helping kids who have these disorders. And I'd like to thank you for your attention. Do you have time for a couple questions?
We have about 5 minutes
Okay.
for questions.
Yes. Yes.
I'm going to wait for that because I know they're recording.
Okay.
I know that imaging data like this is not a way to diagnose individuals, but do these kinds of results influence the writing of new DSMs and the changing of? I know that pediatric mental illness diagnoses the definition can be difficult. How does what you do influence that now or for future versions of the DSM?
Very, very good question. Yeah. So, does what we do influence the diagnostic categorizations in the new iterations of the DSM? And absolutely. I mean, the writing of the DSM is a very laborious and complex process, as you know, and there are a variety of influences that determine what comes out of the various work groups. But, specifically thinking of autism spectrum disorders, they have decided that they're going to eliminate Asperger's disorder, which has become a huge social political issue amongst advocacy groups. But that being said, part of what determined the elimination of that category and just having a broader umbrella of autism spectrum disorders was the fact that when they looked at a variety of studies, including imaging, they found that there was no distinct biologic distinction between high-functioning autism and Asperger's. Okay? So the imaging studies that look at high-functioning autism and those that look at Asperger's were no different. So yes, in fact, data like these are always looked at in terms of DSM categorization.
Hi. So I keep thinking about that picture you showed us of the age of onset of the various disorders and when the brain is developing. Yep. And I noticed that mood disorders and substance abuse disorders are very drastically towards the end of when the brain is developing. Yep. Just what are your thoughts on that, and how is that a sign that brain development doesn't have to do with these things, or are they a sign that it has to finish happening before the onset? Am I making sense?
It's an excellent question, and I don't have a straightforward answer to that. The fact is there's a range of age of onset, and I think that having some of these disorders, for example, anxiety disorder, ADHD, for example, even mood disorders, okay, predispose people to things like substance use, because youth self-medicate just like adults do. But I think the access to substances, just inherently because of laws, occurs a little later. Sometimes kids dip into their parents' liquor chest, liquor cabinet, but they don't have ready access to those things because of the law. And I think that in part influences the age of onset, if you will. Yeah. Yes.
So Jean, this was really excellent, and thank you so much. So I do have more questions about the development, and have people looked at like delaying the growth spurts at that adolescent age to sort of delay the onset of, or stop the onset of some of these diseases, or is there anybody looking at trying to prune away parts of that development or things that happen during that time as a way of stopping the onset of, during the pruning phase?
Yeah.
Could you like could you prune away the disease somehow?
Could you prune away
Yeah,
the disease?
yeah.
Wouldn't that be nice?
That,
I
yeah.
don't know. I think it's such an excellent question. And obviously, wouldn't it be nice, for example, and I have some faith that we might get there one day, that we could use imaging as a little bit of a litmus test to either determine who's really at risk by taking a picture and then figuring out what we need to improve that. There are many things that we can do, for example, non-pharmacologically, that can reverse certain types of things. I'll call upon the major depression literature in that regard, and even ADHD literature. It's pretty clear that there are certain non-pharmacologic interventions that can be helpful. And for example, cognitive behavioral therapy really can reverse major depression. And there have been some nice imaging studies, both in adult and adolescents, looking at the relationship between symptoms and how their brain lights up in response to certain facial affect, and their ability to sustain the correction of the abnormal lighting up of the brain. And it's clear that cognitive behavioral therapy is just as effective and actually may have more sustained benefit than pharmacotherapy. The truth is, most youth who have these disorders require both medication and some sort of therapeutic intervention. But obviously, what an elegant question that taps into the wish. And we're moving there, but we're certainly not there at all.
I actually have a two-part question.
Sure.
You had mentioned about getting a database of normal brains.
Yep.
And I was wondering where they came from. And when looking at those brains, did you find that they were actually abnormal without expressed disorders?
Well, okay. So the multi-site project that we're involved in is a 9-site study, and there are a variety of different groups of people involved. I'll probably not be able to mention all the sites because of my aging memory, but for example, one of the sites is in Hawaii, so it's a national effort, and we collected brains, therefore, from images of healthy kids from all over the country, and all ethnicities, boys and girls, from age 3 to 21, and inherently we excluded anybody who had any psychiatric issue, with the exception of if somebody had a mild anxiety disorder or ADHD, they were considered part of that normal group. So we didn't have psychopathology represented. It's really definitely a normal brain acquisition. Could some of the kids who were enrolled at 3 at some point down the road acquire some sort of emotional behavioral problem? Absolutely. But this was a cross-sectional study, and we're not following them over time. Did we have abnormal findings on the scans? In healthy control studies, about 10, I think it depends on the study, there can be somewhere between 5 to 10% that have mild abnormalities that don't reach the level of clinical significance. At our site, I can speak to our site, we haven't had any clinically significant abnormalities on the scans. So, excellent set of questions. Okay, well, I think we're out of time, and you guys have to go on a tour.